4 months between that. And then at the same time, we have variation in magnitude. If we look at the yearly incidence per 10,000 individuals, and we're looking at states from north to south, you see variation within a given state across years, and then for a given year, a lot of variation. Just to say that the dengue situation across Brazil is not consistent. And so you've seen this slide a couple of times. This is our go-to slide, because this is our goal, our ambition. We want to improve decision support by monitoring and forecasting diseases, using different types of data streams, combining them with all sorts of different modeling techniques in such a way that we can quantify uncertainty. But since there is so much variation, we probably need to think a little bit about how we actually build these models and try and account for the sources of variation. And so what I've been working on is trying to tease apart some of the ecological underpinnings for why this variation occurs, so we can incorporate that into our model structures, therefore improving both predictability as well as interpretability. And so why might this be challenging, or what might be some of the sources of variation of dengue in Brazil? The first is that the epidemiological dynamics are not stable. Even though dengue has been circulating for a while now, you have multiple different serotypes. They've been introduced into different states at different times. And so there's in no way kind of a stable equilibrium here. There's 5 distinct climates, so, and for vector-borne diseases, we know climate impacts transmission. You have the coexistence of multiple vectors of Aedes aegypti and Aedes albopictus, and even though they are similar, there are slight variations in the preferred habitat, their food source, and so where we have overlap of the different vectors, you might see different dynamics. Same thing with multiple arboviruses, there might be competition occurring. And transmission is highly localized, and this was brought up yesterday, where dengue isn't like flu, it doesn't diffuse across the entire state, you have more localized pockets of transmission. And then Marcelo talked a lot about the issues with reporting. So these are kind of the challenges that we have to think about when trying to build a good prediction or forecasting model. And it also points to that building a statistical model might be easier than trying to mechanistically describe all of this. And so thinking about those challenges, these are the questions that I've been looking at to try and build that foundation for how we might want to better improve our prediction models. So first, just asking, how do outbreaks vary across time, space, and scale? I alluded to this a little earlier. And then, which environmental and socio-demographic factors are associated with specific properties? So getting at where might we see signal in data that can help us? And then finally, how do we translate what we know to something tangible for public health purposes? Okay, so I'm going to go through each of those 3 questions, the results that I have so far. Okay, so if we think of a multi-year time series, such as this for Sao Paulo, there are many ways that you can describe the behavior, the typical behavior, besides just looking at the entire time series. So thinking back to those public health, or those objectives that the CDC has for forecasting flu, onset, peak, all of those things that are very quantifiable, we could ask things like, how long is the typical season in a given state? Or furthermore, what is its shape? Do you see kind of flat transmission throughout the year, or is your outbreak usually peaked, concentrated in a few weeks or months? You might also want to know, how consistent is the onset? How consistent is the peak? Do you expect the dengue season to start the same week every year, and that's something that's really reliable, or can it vary within a couple weeks, a couple months even? And then finally we see that there's a lot of variation in the magnitude, and that's also something very relevant to these forecasting or prediction forecasting systems. At the same time, It might be possible to use information from a neighboring state or from even a state quite a ways away. And so we can define properties that are more relational between two different locations. So here, looking at Sao Paulo and Tocantins. If we look at the number of cases, and this is normalized, so they're on the same scale, because Sao Paulo, absolute numbers, much higher dengue burden. You can ask some questions such as, how in sync are these two states? Are they rising and falling at the same time? And if not, is the lead and lag consistent? Is that something that's also reliable? Do you always see a 3-week lag between the two? Or in some cases, is there some variation in that property? And then we can also ask how similar are the relative magnitudes? Do large outbreaks occur in the same year? Do small outbreaks occur in the same year? And so for Sao Paulo and Tocantins, the small outbreaks are actually in different years, and the large outbreaks in different years. So they're not necessarily in sync in terms of their magnitudes. And so we took this approach of thinking about these different properties of a time series, and one way to kind of get at these is you can do decomposition of a time series. So if we think of this as the original data, there is this trend component, which is capturing kind of long-term increases or decreases in the number of states. There's the seasonal component for something like Brazil or dengue, we know that's occurring, and then you have some sort of randomness. So these are, this is the kind of approach we took for looking at the different time series. We decomposed time series from at each state and each mesoregion level from 2010 to 2016. So that's the data we analyzed. Okay, and so we defined 4 properties. 2 of these are intrinsic, meaning they're just talking about the specific state or mesoregion itself. And the first one is what I call intensity, which is a little bit confusing because it's not talking about the actual absolute number of cases, but it's rather talking about the shape. And it's saying how intense is that peak. You can think of this as an entropy measure as well. How kind of diffuse is it throughout the season, or is it really concentrated? So you can see that Sao Paulo has a higher intensity measure. Its peak is usually a little bit sharper. The next metric we looked at was the seasonality intensity, and that's basically saying how well does it fit a 52-week cycle? How kind of consistent, how reliable is its rising and falling over the 6-year period? And so Sao Paulo, again, has a stronger seasonal signal. In terms of these extrinsic outbreak properties, these are those pairwise ones. The first one we looked at was the trend. How well correlated is the trend? And then the second extrinsic feature was the timing. So how well correlated is just the timing component, separated from everything else? And so you can think of this as a correlation. So here, this is the correlation value between these 2 states. Or you can capture the average lead or lag, so whatever measure is more useful. Okay, so we calculated these 4 properties for every state and every mesoregion. And what we found is that they are independent properties. They have their own patterns, and so they are kind of useful quantities in themselves if you want to characterize outbreaks. So this is the intensity pattern that you get at the mesoregion level. And sorry, this is a little hard to see, but yellow indicates low values, and purple indicates high values. So that's where your outbreaks are like this. And then seasonality, high values mean they're very consistent, the timing is very reliable. And so we can see between intensity and seasonality that they're not the same. They're giving you different pieces of information. Here in the north, you have flatter outbreaks consistent with year-round transmission, but their timing is not very reliable. Here in the south, you have very sharp peaks, and their timing is not reliable because they're probably, from what I understand, they're probably more related to random importation values and there's just a lot of stochasticity more in the model. We found the same thing if you're looking at correlation and trend and timing. These are also different independent properties. So here on the y-axis, this is the ranked order by mean pairwise correlation. So in general, how well correlated is a state with the rest of the country in terms of timing, and then in terms of trend over here. And so the state that is on average the most well correlated in terms of their timing is Minas Gerais. Close? Okay. And so you can see that this does have a distinct order, but then the order, if you're looking at trend, is quite different. There is really no one-to-one matching, and so these are different features you might want to build your prediction models around. Okay. So that is.
Can you talk about the connectivity? Or what?
What are
Yeah.
those lines?
Oh, the lines, they're just trying to, so you can better visualize that. So Minas Gerais is number 1 here, but then it's number 11 in terms of trend. And so there's, you don't see, what the point is that you don't see straight lines across. They're very slashed. Diagonal.
Did you try to make some kind of cluster analysis, take these kind of factors that you are from each state and try a cluster analysis to see if they are forms like groups?
Yes, I'll get to that. Yeah, exactly. But this motivated this next question about which environmental and socioeconomic factors can explain these outbreak properties. And 2 questions here really. Does the same, do the same factors explain the same, all of the properties, or are there specific factors associated with different properties? And thinking about the influence of, or not the influence, but the decision of where do you actually want, at what geographic scale do you want to build these prediction models? Do you find different variables, whether you're trying to explain state-level properties versus meso-level properties? And so as Carrie mentioned earlier today, we have a range of different variables that we've collected over the same time period from 2010 to 2016. We have remote sensing, more weather-related data, and then more socioeconomic data that we know might influence the interaction between mosquitoes and humans. And so Carrie went over these, but a lot of these remote sensing ones are getting at vegetation and precipitation levels. And then we have mean temperature, relative humidity, temperature range, and then the socioeconomic factors. And just as we decompose the time series into these different components, you can do the same thing with environmental time series, right? And so if we want to, if this is our target eventually, if we want to say how well does a given remote sensing variable explain this pattern of dengue seasonality, it might be annual average vegetation levels, kind of just an understanding, a summary statistic of the baseline, or it might be the actual seasonality in how vegetation levels change. Is the vegetation consistent, or are changes in the vegetation consistent year to year, or is that a chaotic feature? And so just as we decompose the outbreak time series, we also did the same for our environmental time series and considered that as a potential signal. And so to identify which factors were important, we took a lasso regression approach, so to help try to reduce this variable selection space, and performed k-fold cross-validation using 80% training and 20% testing data. And to get a sense of how stable these signals were, we performed this 100 times and then looked at the distribution of results from that. And we measured error by looking out of sample relative mean squared error and Pearson's R correlation. And similar to how Carrie explained, different subsets of data might be informative of different geographic locations. We tested, again, do different subsets or different types of environmental and socioeconomic data. Does that make a difference in your performance? So we considered fitting models just based on average summary statistics. We fit models based on the environmental data, but in terms of the intensity or the seasonality. And then several other combinations, including the inclusion of different socioeconomic data. So these are examples of the different types of results we might expect. Okay, so I'm going to go through how well, so again, what we want to assess is which properties are explainable by environmental and socioeconomic factors. Are they the same across properties, and are they the same across scales? Okay, so looking at seasonality, so this is again, how consistent is the timing year to year. And looking at the out of sample relative mean squared error, where you want lower. The blue bars indicate the results for the mesoregions, the brown indicates the state level, and then the bars are at the median, and the error bars are the interquartile range over the 100 fits. First, we can see that the state level usually has lower error, so the models are better at predicting state level properties. And there's not, though, a huge difference between the data subsets. A little bit here where just these environmental-based factors seem to be the most useful at predicting seasonality, but perhaps there's a lot of redundancy. If we look at intensity, the error here in intensity is much smaller than seasonality. And so that's to say that intensity is the shape of a dengue outbreak is more explainable by socioeconomic or environmental factors. And I say both because regardless of what type of data you're trying to fit it with, you get about the same level of performance. Looking at timing and trend, now this is on a different scale than this one, but we can see that timing, again, there's just not a huge difference between the different variable subsets, but here the state level actually tends to have a higher error, so predicting or explaining mesoregion timing is better than at the state. And then trend, we can see, has a very high error compared to all of the rest of the properties. And so trying to say something about patterns in relative magnitude, we can't do that with environmental and socioeconomic data, and that makes sense because the magnitude is probably going to be related to more epidemiological factors such as serotypes and susceptibility levels, and not so much what's happening in the environment. So if we look at the Pearson's R correlation, we see the same story. Sometimes these error metrics can give you slightly different results, but here we see it's pretty consistent where state level timing actually has the highest correlation between our predicted and observed values. Intensity is about the same. We can explain about 80% of the variation. And then seasonality follows, and then trend, we're only explaining about 40%. Okay, so what were the top variables across different properties? Were they the same? Were they the same across different scales? We found, if you're looking at intensity, that the variables that seem to come out as important in the models were the annual burn ratio and the annual temperature. So the burn ratio is a measure of drought sensitivity. So it's measuring the level of precipitation in the environment, and then temperature. And then in terms of the mesoregion, you see the same variable is included, the normalized burn ratio and the temperature, but the best model actually came out when you were using the shape of these variables and how the shape of the normalized burn ratio compared to the shape of the dengue outbreaks. And then we see the rest of these are, again, related to water, the percent cloudy pixels, the water content in water bodies, and the vegetation indices. So these explain the typical shape of a dengue outbreak. If we look at seasonality, the best models had some similar and some different variables. So the top one at the state was the water content in bodies, and then you see this burn ratio again, and then relative humidity comes in. And so that makes sense as something that's controlling the timing. We know humidity is important for mosquito development. And then again at the mesoregion, we see that these kind of more higher, or these more complicated, sophisticated measures are included in the top models. You're not just using kind of summary annual baseline statistics, you're saying something a little bit more complex about the time series. Okay. And then here are the results, the top variables for timing and trend. Timing at the state, it's unsurprising that distance ends up being the most significant variable there. The further you are in terms of states, the less likely your timing is probably going to be in sync. And that's at the state level. However, at the mesoregion level, distance wasn't as important actually, and it was more in terms of environmental synchrony that determined whether or not two outbreaks were the same. So this is just again saying that depending on your level of analysis, you might get different results. Interestingly with trend, even though these models didn't explain the variation in trend very well, they do come out with different variables, and the trend tends to be more explained by socioeconomic factors such as household density, the percent rural population. Okay, so some takeaways from this point. Outbreak shape is more predictable than season target. So kind of the practical element that falls out of this is that building statistical models that might capture the shape might be a more feasible kind of first step than other elements of the dengue time series. When we looked at mesoregion, we believe the mesoregion scale or unit might be better at capturing actually what's going on there because you're not aggregating multiple different dengue outbreaks into one. And we found that timing and shape were explained by remote sensing indices. Trend was explained more by socioeconomic. And so we're still working on this analysis, and we're trying to incorporate more connectivity measures in addition to just distance, because that was really the only connectivity measure we had in that analysis. So we are looking at bus, airport, and also Twitter data as a proxy for how connected two different locations are. Inspired by this workshop, this is all kind of more correlational, but we haven't done actually causal analysis, so that would be interesting. Okay, but getting to this final question of now how do we use this information for prediction and public health purposes. And so we were inspired by this sort of heat map if you're just looking at the correlation in epidemic timing. And so here on the x-axis we have states, north to south, and then north is here and south is here. And you see that clusters of high correlation in terms of timing are geographically structured. So there is structure in these different properties individually that you can probably include into some sort of statistical prediction model. And so as a first pass, we applied k-means clustering to the individual properties to see is there evidence for clustering. And why this might be useful is instead of trying to include every other location in a prediction model for a given state or mesoregion, you can narrow your search and you can pull in the information that is most relevant. And so this is more of an efficient way of exploring your model space. And so we found evidence that there are 4 distinct timing clusters, meaning clusters in this mesoregion here, they have similar timing to each other, and they also, their distribution of timing to the rest of the country is also similar. And so we can see that the timing clusters fall mainly across this longitudinal gradient from west to east, which is similar, as I said, that the timing goes like this. And if you look at the distributions in terms of the number of weeks leading other mesoregions, mesoregions in this cluster have the highest lead, and they look significantly different than the number of weeks in this cluster leading the rest of the country, and so forth. And so in terms of the 4 properties, timing showed the highest evidence for this clustering structure. Trend did not show any. Intensity showed a little bit, and seasonality not really that much either. And there was little evidence at the state level, but there was a high evidence at the mesoregion level for clustering in terms of these properties. If you don't want to consider each of these properties separately, you can consider all of them together. And so this is looking at the values of intensity, seasonality, timing, and trend, and clustering on all of them together to say, are there distinct outbreak types across the country? And we found evidence that yes, there was. In this case, we chose cluster K of 5, and so these are the different types of clusters you would find. And we can look at what the average value of these different properties are for these clusters. So here in the south, in this green one, you're typically seeing outbreaks that have a high intensity value. They're really sharp, but their seasonality is low, so they're not very consistent in terms of their timing, but they tend to be correlated in terms of their timing and trend. Alternatively, you can look at this yellow one and see that it has lower values of intensity, but its seasonality is higher and its trend is the highest. And this is, I think, Munich Gerais is in this area, so that makes sense. And its trend is a little lower. And so we think something like this sort of analysis will be useful for building better prediction models around. Because it's based in more of a data-driven way. Okay, so concluding thoughts. Intensity and timing are reliable signals. And this is despite some of these challenges that we were talking about. So these are properties that we can explain, that we don't need to kind of consider more complicated mechanistic dynamics. We found evidence that thinking about dengue on the mesoregion level makes more sense than the state level. Maybe it makes sense to go even further down. We haven't explored that yet. And similar to what Carrie said, we do find continued evidence that it is worth the effort of collecting different types of data streams and testing them, that they provide different information, and at different locations you need that. And I'd like to say thank you. And this is our team at Los-Alamos, and we've gotten very useful advice and discussions from our collaborators at Harvard, Northeastern, and Peers. So if you have any questions, I'd be happy to answer.
Thank you. So questions and comments?
Yeah, I was wondering if it is the temperature and these ideal conditions for the mosquitoes that are driving the dengue dynamics, then those should be more predictive of mosquito dynamics than they are of dengue dynamics. Do you have any data, or are there any data, longitudinal data on biting rates, or I know these days you can have a look at people's serology to see if they've had a recent bite from a mosquito. Is there any data like that you could use?
I'm not sure. Marcelo, is there any data? I don't think so.
I don't think there is.
Yeah.
It's not available.
But you could, what about with the new technology for people having a serological reaction to the mosquito bite? Would that be easy to collect? Or not?
I really don't know. What I can say is that as of now, the data across the country for related to mosquito presence or abundance is
Yeah, much less. Yeah.
hard,
Yeah.
if at all.
questions? People don't ask questions in classrooms.
Yeah.
No, they
Yeah, also after lunch.
We need the coffee.
Huh?
We need the coffee.
So, okay.
Where are they listening? Is for the cluster, because the
Oi.
heterogeneous.
Hier
Yeah,
is dat computador.
so that's kind of interesting.